Good. Hello, everyone. I hope you had a good lunch. Happy afternoon. I'm Ashley Elgin, a benthic ecologist with the NOAA Great Lakes Environmental Research Laboratory, and I want to acknowledge my co-authors Paul Gleishaw, also of NOAA GLERL, and Rachel Orzakowski with the Grand Valley Annis Water Resources Institute. We are going to talk today about dreissenid mussels with a focus on Thunder Bay and Saginaw Bay for population assessments, but then we're also going to look at lake-wide body condition, how the mussels are doing personally, not just as a population. I first want to quickly acknowledge that we got to work with a lot of great people in the course of this study in 2022. A few highlights is thank you for EPA and Great Lakes Restoration Initiative for support. And also, saviors of this year were that DFO and the Canadian Coast Guard came and helped provide us with a vessel when the EPA ship couldn't make the survey, and my crew and co-authors had a great time being on the survey. So to connect to the CSMI priorities and what motivated the work, there are priorities under both habitat and native species as well as invasive species, just to say we need to keep track of what's happening with mussels. They're an important stressor in the system, they're an important part of the food web, so let's be following their populations. Along these lines, a group from NOAA and we had our Cooperative Institute partners and others, we conducted fine-scale spatial studies and looking at the lower food web and primary productivity. I was in charge of the benthos sub-project, and for that we looked at what I'll present today is the whole lake body condition and the abundances in the bays, but we also collected mussels for mercury analysis, and that was collaboration with USGS, and we collected water samples for eDNA, that's a University of Michigan partner on that. So we tried to get as many samples of opportunity as possible. It was a good coordinated effort. If you're interested in what's happening with the mussels in the whole lake, Jim Watkins said he presented on that earlier and showed some of the whole lake results that are led by the Buffalo State University's Great Lakes Center. And when that report's available, I put the link, so if you're interested in seeing their whole lake report for benthos, you can go to that page and find the report. So in big picture, in my lab at NOAA, we've been doing these lake-wide assessments with the CSMI cycle of quagga mussel body condition. These are all the sites that we've hit since 2015. Many of these sites have been hit 2 times since 2015, and today I'm going to be focusing on the Lake Huron sites that are circled there. I doubt anyone will be able to jot down this DOI this quickly, but if the presentation's available and you can see it, I just want to say this is a record that we add more data to every CSMI cycle. Our Ontario 23 data is already up there. So if you have interest in shell and tissue metrics and this kind of coverage across all the lower lakes, or all lakes except for Superior, please check out this data set. So jumping right into what we found for the mussels body condition, and here we're expressing it as the amount of tissue relative to a given shell length. And first we visualized this as a series by these regressions, and we color-coded and did analysis by depth zone. Mussels certainly respond, and they have different environments by depth, and their population trajectories really depend on depth. And so in this figure you can see this represents each dot is a mussel. So I say how the mussels are doing individually and personally. I very much want to see how the mussels are doing at each of the depth zones. How does that match on to how their populations, what direction their populations are going? Here, the inset figure shows the least square means as a result of ANCOVA, and we did paired comparisons, and from that you can see that the mussels in the black group, that's the less than 30 depth zone, they are relatively heavier and in a class of their own compared to the other depth zones. And then the deepest mussels, as well as one of the mid-depth zones, were intermediate, and the skinniest mussels, tissue-wise, are in that 51 to 90 depth zone. Interesting is when you look at mussels in the 51 to 90 depth zone, this is a zone of greater density for mussels. So it could be that food resources are limited there, and there's more local competition for the food. And that's maybe one way to say where the C group is more depressed. So I'll put this in perspective in a few ways. First, I'm going to compare 2022 to 2017. So the 2017 values are the darker bars, and 2022 is the lighter bars. Doing a two-way ANCOVA with these, we found that the depth zone is significant when you look at both of those factors simultaneously. However, year is not significant. And you see there isn't too much difference between the pairs of bars within each pair. There was a depth by year interaction, however, and I think that can be seen here at the less than 30 meter sites. This is where mussels were already the beefiest, and they became beefier. And then in the zone where they were the lightest, they became lighter. And those were the trends over time, and those conflicting trends led to an interaction. In the other depth zones where the mussels are intermediate in condition, there's negligible change between the 2 years. So how does Lake Huron compare to the other lakes? These are all data that we have from that archive data set showing all those stations across the Great Lakes Basin. I just threw a line across for visual comparison. That line doesn't have meaning besides it's around 6 milligrams of ash-free dry weight tissue for a 15 millimeter mussel. That's how these are all standardized by 15 millimeter mussel. So across all lakes, you'll see that the less than 30 meter mussels in that shallow zone are always the heaviest. They always have the beefiest amount of tissue. It's especially the case in Lake Erie, but in general, the more shallow you go, the more robust mussels you're going to have. It's warmer, it's more productive, and that all fuels a lot of energy that can go towards tissue and shell growth. Kind of across the lakes, you tend to see that the next robust mussels tend to be the deepest ones, which is interesting, but they're in a low food environment, but their populations are generally more sparse and spread out. And in the mid-depth zones will be intermediate. Of course, we always exceptions to that. So now I want to transition away from thinking of the whole lake to what's happening in Saginaw Bay and Thunder Bay, where we focused our efforts in 2022. So I'll do that by just pulling out what was happening condition-wise at those 3 sites. These are presented the same way as before, and looking at using the ANCOVA, we are looking at least square means with paired comparisons. And Thunder Bay, which is up in the north, we have one Thunder Bay site. The mussels were just as beefy as at one of the Saginaw Bay sites, Saginaw Bay 15. And both collectively, they were significantly higher than Saginaw Bay Site 5. When I look at this, and I want some of you looking at this and you're thinking the Saginaw River plume is coming out, one of those sites in Saginaw Bay is going to be more in the plume, maybe getting more influence from the river, and that would be the 15 site where we see heavier mussels. So this is a transition to, this is what's happening with the mussels' growth, where they're putting energy towards growth, and they're clearly supporting tissue growth. In general, these very shallow sites are higher than shallow sites that are in the main basin. Over time, I forgot I had this one in here, if we look at what's happening between, in the reddish bar, 2017 to 2022, and the blue bar, the ranking among those 3 stations is identical, and all 3 of those stations increased. And that follows that trend of the nearshore mussels are doing a little better than they did 5 years ago. So now looking at the surveys. We did a ponar and a diver survey in Saginaw Bay. We have a mixture of substrates there, and it ends up being 10 ponar stations, which are in orange on this map, and 3 diver stations in green. We assessed the mussels. We looked at we counted them, we weighed. This was total mussel wet biomass I'll be presenting, and we also distinguished between quagga and zebra mussel species. Because this is an area where we do get coexistence. A lot of the surveys that I do in other areas, you never see a zebra mussel. We got to see a good number of zebra mussels here. So I'm going to I decided instead of showing you a mean for the bay, I want to show you site-by-site data. And the error here indicates variability among ponar replicates. We have 3 ponar replicates or 3 quadrats at each site. And what just jumps out very immediately here is most of the sites are low to absent mussels. You have a few intermediate, which on the bottom are in the green bars, and 1 high site. High site is SP15, and from a previous slide, that was a site where the mussels were personally doing very well. This, the figure here shows, I'll reach, pretty much all the way through. All these dark colors indicate where mussels were absent or very low numbers. We had a whole day on the waters. The last time I was at this hotel was when I was here to do this survey. We had a whole day on the water where every ponar that came up, we're like, well, where are the mussels? Based on 2017, we expected to see some, but the mussel numbers were really reduced all throughout the center of the bay. Now jumping up north to Thunder Bay, we had 5 stations, all of which were sampled with divers using triplicate quadrats at each station, and handled the samples in the same way, quagga versus zebra, density and biomass. I put the same color coding on this map, which connects to the density values. Thunder Bay is also patchy. We had 1 station that was way out at the end, station 3, that had the highest density and the highest biomass, and all the other stations were low levels of density and biomass, and 1 site had no mussels at all. So now I'm going to throw together a lot of summarized data from the last few slides and bring an additional year for comparison. So let's digest this figure here. We'll start out on the left side, look at Saginaw Bay with the darker color being 2017, the lighter color being 2022. We see a huge decrease in especially density in Saginaw Bay. This was very much driven by a lot of very small mussels in 2017. And we didn't have as many small mussels, but just also didn't have nearly as many mussels in 2022. The change in biomass is more negligible, and that's, again, because it's a different size distribution in each of those collection years. I'll highlight that the percentage at the base of the bars, that shows what percentage were quagga mussels. As far as quagga mussels go, the percent increased or stayed the same over time in both Thunder Bay and Saginaw Bay, density and biomass. Another thing I'll point out here is in Thunder Bay, the density and biomass declined more in step with each other to about 1/3 of what it was 5 years ago. See, these declines and about 1/3 for both. And then what's interesting is the biomass far exceeds that in Thunder Bay than that in Saginaw Bay. And one way to look at this is I just calculated average weight per mussel. The mussels in Thunder Bay are much heavier because they just are, it's a larger size distribution than they are in Saginaw Bay. And you can see that in Saginaw Bay, on average, the mussels got bigger compared to 5 years ago. But in Thunder Bay, not too much of a change, but they are a little bigger. I also broke out here between quagga and zebras, and they have comparable average weights in Thunder Bay, but Saginaw Bay has, they're a little lighter, but they also tend to be smaller. So to wrap up here, I just want to say overall the densities in Saginaw Bay are a fraction of what they were at the peak in the 90s. In the 90s, that explosion of zebra mussels just created historic highs in Thunder Bay, or in Saginaw Bay, and the values are a little lower, and depending on how you measure it, than 5 years ago. But that's just 2 measurements 5 years apart. It's hard to get strong trends from that. But in perspective, total dreissenid numbers is way down from what it was in the 90s. The populations are highly patchy, and that's been the case for the last 20 years in both locations. Work by Pothoven and Nalepa looking at whitefish starting to use dreissenids as a food source showed that they were, even though the dreissenids were extremely patchy, whitefish were incorporating them, especially the larger whitefish, into their diets. Another thing we noticed is that in 2017 and 2022, we were sending divers down to stations that they didn't need to dive anymore because we could have just done a ponar at those sites, and that's a change from the past. And we've had some change in water levels, but I just had a thought: is there increased sedimentation into that area, the inner part of Saginaw Bay, and maybe it's creating a substrate that's not as favorable for mussels as having more hardscape than we did in the past. I'm interested to hear anyone's thoughts on that who's more familiar with sedimentation in the bay. So ultimately, I'm thinking, is there more biomass available for fish predators in Thunder Bay? Population still being patchy, but it's a lot of good-sized mussels that are available there. We know that fish are utilizing mussels as a food source and thinking of them too as they're both the stealers and the providers of energy in Lake Huron. So with that, we have some time left for questions. Here's my email, ashley.elgin@noaa.gov, if you think of anything after today. Thank you.
Thank you, Ashley.